Nanoparticles are microscopic particles with at least one dimension less than 100 nanometers. We made iron oxide nanoparticles, which are magnetic. These particles are used as contrast agents for magnetic resonance imaging, or MRIs. The nanoparticles were made by mixing iron 2 and iron 3 in an ammonia solution. They were then tested on brine shrimp to see what the toxicity would do to them.
In class, we're learning about the properties of the atom, which can be studied using nanoparticles. In the real world, nanoparticles are used for targeted drug delivery and contrast agents.
Our initial hypothesis was that the citrate would increase the magnet properties of the nanoparticles as well as increase the toxicity of the nanoparticles. This is because the nanoparticles have a greater surface area to volume ratio. With a greater area for bonds to form, the strength will increase. The other is because citrate is used in many medications. So mixing a type of things as used in medicine with iron oxide, which is a salt, is not a good combination. Salts and medicines typically do not mix well. In the end, the citrate actually did not affect the magnetism properties of the nanoparticles, and it actually decreased the toxicity of the nanoparticles as well. On day 1, we mixed iron chloride 2 and iron chloride 3 with ammonia to make the nanoparticles. For our group, we made the control, so we just added the designated amount of water. But for citrate groups, they added the citrate and the amount of water that was given to them. Both of us stirred on a hot plate for 30 minutes at 80 degrees Celsius with a stir bar.
After 30 minutes, we took the beaker off the hot plate and used a magnet to draw the nanoparticles to the bottom. We then poured out the excess ammonia into a waste beaker, with the nanoparticles still at the bottom. The nanoparticles appear white here, but they were actually a dark brown, blackish color. We then added 50 milliliters of water, swirling for about a minute to redisperse the nanoparticles. We then used the magnet to keep the nanoparticles at the bottom, pouring off the water. We did this 4 times, removing the water each time. Then, once the nanoparticles were clean, we added 100 ml of water to the final solution. In the lab, we used Beer's law. Not this one, this one. Beer's law states that the light absorbency of our solution is directly proportional to the concentration of the solution.
In class, we took concentrations of known solutions and used Beer's law to measure the light absorbance with the logger pro of what each solution was. We then used Beer's law and found our unknown concentration of our nanoparticles using the slope, the y-intercept, and the absorbance given to us on the logger pro.
The best contrast agent should be the citrate solution because it lowers the toxicity. However, based on the conclusions done in the experiment, the control has the lower T1 value, so it would be the better contrast agent. The T1 value for our experiment was high compared to the other control values.
We hypothesized that the lower concentrations of nanoparticles would have less of an effect on the brine shrimp, and that the higher concentration of nanoparticles would have a more damaging effect on the brine shrimp. The results taken 24 hours after our experiment showed that the nanoparticles had a very deadly effect on the brine shrimp, and we looked at the brine shrimp under the microscope, and the particles had all been absorbed by the shrimp, and all the shrimp were dead, which therefore proved our hypothesis to be incorrect.
So we use brine shrimp in this lab because they're filter feeders, so toxins get into their system a lot faster, so it's easier to study the LC50 and stuff like that. So,
let's do this. So our experiment tested the effects of different solutions of nanoparticles on brine shrimp. This applies to the LC50, which means lethal concentration of a solution for 50% of the population over 24 hours. Our experiment tested this by placing 10 shrimp in 4 different solutions in 1 control and waiting 24 hours before observing how many shrimp were still alive in each group compared to the beginning.
the concentration, we concluded that none of them could be safe due to the control brine shrimp of just seawater died, as well as all of them through the concentrations died, 1 to 1, 1 to 10, 1 to 100, 1 to 1000. Couldn't conclude a safe toxicity due to the sense that our control died and the rest of them died as well. So from that, we couldn't tell where if the nanoparticles were at the lowest, if they were better for the brine shrimp, or if they were at the highest, was it better for the brine shrimp? Prior to the experiment, like, we would have either changed the species that we were using for brine shrimp, or we could have actually taken the time to figure out the toxicity prior to the experiment itself.
This could be used in humans, and that way the brine shrimp can be observed in several environments to find out the various water qualities and what's affecting the environment and how it can be fixed.
The applications that our experiment would have on humans would be kind of understanding how the nanoparticles affect humans and how an overdose in nanoparticles on humans how we would react to that type of exposure to different solutions of nanoparticles like our brine shrimp were exposed to.